My name is Hanqian Zhang. I'm a PhD student at Uppsala University in Sweden. Thanks very much for ESDR, Lily, and Hood Foundation providing me travel grant to attend this great ESDR conference in Salzburg. Also, thanks very much for giving me opportunity to present my work. My study is ichthyosis patients with TGM1 mutations show aberrant transcriptomic expression with a focus on the expression of ARCI-causing genes. Autosomal recessive congenital ichthyosis is a group of rare monogenic skin disorders. ARCI is characterized by impaired skin barrier function causing hyperkeratosis, which results in a dry, thick epidermis and scatty skin. About 10 genes have been reported as causative genes in ARCI. Among them, TGM1 is the most commonly affected gene. Most ARCI-causing genes are involved in isoceramide synthesis. As you know, epidermis has 4 layers. In the uppermost layer, stratum corneum, there is a structure called corneal site lipid envelope. It is a key structure for skin barrier function and ichthyosis pathogenesis. It is produced from the proton bound isoceramide to the cornified proton envelope, catalyzed by transglutaminase TGM1. And most other ARCI-causing genes are involved in different steps of isoceramide biosynthesis, as shown in this yellow box. And the underlined genes are ARCI-causing genes. However, the effect of mutation in TGM1 on the expression of other ARCI-causing genes remain elusive. Therefore, we performed a micro-study to investigate the transcriptomic expression differences between controls and the patients. So then we take 2 skin punch biopsies from each subject of 4 healthy controls and 5 ARCI patients with TGM1 mutations. And 1 skin biopsy is used for RNA extraction, the other is used for frozen and for cryosectioning. The RNA sample later is used for microarray analysis and qPCR to validate the array result. And we also can do immunofluorescence on cryosections of the controls and patients to validate the array result in protein level. And here is the patient mutation information. So based on the microarray result, we draw a heat map to show the ARCI gene's expression profile. In this heat map, we can see 4 healthy controls clustered together and 5 patients clustered together. And the red color means increased or higher, relatively higher expression. Green color means relative lower expression. And those ARCI genes are increased in the patient compared to the controls. Then we did qPCR to validate array result. Then in this qPCR graph, we can see the white bar control and the gray bar patient. The most ARCI genes are significantly increased in the patient compared to the controls, which corresponds very well to the array result. We also did immunofluorescence staining of 3 ARCI proteins on cryosections of control and patient skin. Then we take 2 examples. One is control 4, the other is patient 4 to show the staining result. We also used a new layered analysis method to analyze the staining result. So the layers from top to the bottom in the image is correspond to the intensity measurement from the left to the right in the graph. And the gray area indicates the stratum corneum in the image. And then the granulosum spinosum is going to the right. So then from the images and the graph, we can see the increased O3 proteins expression in patients compared to the control skin. Then we conclude that microarray data show several epithesis genes are upregulated in the patient's skin compared to controls, which is verified by qPCR and the immunofluorescence staining partially. This indicates a feedback induction of isoceramide biosynthesis to compensate for impaired skin barrier function. It seems that the skin patient tried to produce more isoceramide to restore the skin barrier function. The study is supported by Swedish Research Council and the Hood Foundation. Thank you.